Greetings everyone. Welcome to my YouTube channel PathQuest. I am Dr. Gauri Medkar, Professor and Consultant Pathologist. This is the third lecture in the lecture series Cell Injury. I will be discussing about classification of morphological forms of cell injury, differences between reversible and irreversible cell injury, which include biochemical changes, ultrastructural changes which are detected by electron microscope, light microscopic changes, and gross changes. So let's begin. The classification of morphological forms of cell injury include various types or mechanisms of cell injury, and this is the table which is showing different nomenclatures which are used for different mechanisms or types of cell injury. Let's see these one by one. So reversible cell injury nomenclature used is retrogressive cell change or cellular degeneration. For irreversible cell injury or cell death, the nomenclature used is necrosis. This usually indicates unorganized or disorganized type of cell injury leading to cell death. The nomenclature used for programmed cell death is apoptosis, which is an organized form of cell death. The residual effects of cell injury are known as subcellular alterations, which result in alteration in milieu interior of the cell. However, the cell is still able to maintain its function. Deranged cellular metabolism. The nomenclature used is intracellular accumulation of lipids, proteins, and carbohydrates. These are usually seen when the organs which are associated with the metabolism of these substances are injured. Example being liver. The after effects of cell injury and necrosis include gangrene and pathological calcification. So these are the various nomenclatures which are used to describe different mechanisms or morphological forms of cell injury. So let's just revise what are the changes that occur in the cell and the processes that the cell goes through when it is exposed to either stress or any injurious stimulus. So when the normal cell is exposed to a stress like altered or increased functional demand, it undergoes cellular adaptation, which include atrophy, hypertrophy, hyperplasia, metaplasia, and dysplasia. Now, in these situations, when the stress is removed, normal cell structure and function is restored. When the cell is exposed to mild to moderate level of stress, then it will undergo reversible cell injury in the form of degeneration, subcellular alterations, and intracellular accumulations. In this situation, if the stress is removed, the cell will heal back to normal by repair and healing. If the cell is exposed to severe and persistent stress, it will result in irreversible type of cell injury and cell death. In this picture, we can see different changes that can occur in the heart when it is exposed to different type of stimuli. So this is the normal myocyte or heart muscle cell, and this is the gross photo of the transverse section of the heart. So this is the left ventricle and this is the right ventricle. Now when this normal myocyte is exposed to increased demand, then it tries to adapt itself and it increases in the size. That is called as hypertrophy of the myocyte. Increase in the size of the cell is called as hypertrophy. Now, when there is hypertrophy of multiple myocytes, it results in increase in the muscle mass of the heart. So, we can clearly see the difference between this muscle mass of the left ventricle and increased muscle mass of this left ventricle. So, this is called as left ventricular hypertrophy. Now, when the heart muscle is exposed to injurious stimulus which is mild, it results in reversible type of cell injury where there will be changes in the cytoplasm which include cytoplasmic inclusions, etc., and changes in the nucleus which include chromatin clumping. When the myocyte is exposed to severe injurious stimulus, which it results in cell death, which includes loss of cell organelle and permanent damage to the nucleus. Now, when the multiple myocytes are damaged, it will result in a large area of necrosis, and that is represented by this gray-white area in the left ventricular wall, and it is called as myocardial infarction, and in layman's language it is called as heart attack. Now let's see how the cell injury progresses from reversible cell injury to irreversible cell injury or cell death. So the first changes that occur in the cell when it is exposed to any injurious stimulus are the biochemical alterations. These can be reversed back to normal if the stressor or the injurious stimulus is removed. Now, if the injurious stimulus still persists or if it is very severe or strong stimulus, it results in ultrastructural changes in the cell. And then, if they are still persistent, then it results in changes that occur at larger level, which can be seen under light microscope. And if the multiple cells are affected by these injurious stimulus, then it results in affection or damage to the organ as a whole, resulting in gross morphological changes of cell injury. So what determines whether the cell injury is going to be reversible or irreversible type of cell injury? In other words, what determines the point of no return? The factors determining these are not clearly defined because there is interaction of multiple factors which ultimately define the outcome. However, the irreversibility is defined by two main features. These include inability to reverse mitochondrial dysfunction and profound disturbances in the membrane function. Let's see the differences between reversible and irreversible cell injury. The first change that occurs when a cell is exposed to injurious stimulus are biochemical changes. So in reversible cell injury, there is loss of ATP, whereas in irreversible cell injury, there is irreversible mitochondrial damage. In reversible cell injury, phospholipid breakdown and phospholipase activation is seen, whereas in irreversible cell injury, massive peroxidation due to uncontrolled chain reaction is seen. In reversible cell injury, depolymerization of actin occurs, whereas in irreversible, cleavage of CSK proteins by proteases occurs. In reversible cell injury, increased reactive oxygen species occur, as against irreversible cell injury where uncontrolled reactive oxygen species generation accompanied by inflammatory response is seen. In reversible cell injury, release of calcium from storage site occurs, whereas in irreversible cell injury, because of membrane damage, there is uncontrolled calcium influx. In reversible cell injury, there is just altered metabolism, whereas in irreversible cell injury, there is loss of amino acids. Let's see what are the ultrastructural changes seen in reversible cell injury. So in reversible cell injury, we see there are changes that occur in the cytoplasmic membrane. So these include cytoplasmic blebs formation, whereas in irreversible cell injury, there is severe damage and defects in the cell membrane. So in reversible cell injury, there is just bleb formation or outpouching formation in the membrane and aggregation of intramembranous particles. Whereas in irreversible cell injury, there are defects in the cell membrane. In reversible cell injury, there is generalized swelling of the cell. Then there is autophagy by the lysosomes, clumping of nuclear chromatin, endoplasmic reticulum swelling, dispersion of ribosomes, mitochondrial swelling, small densities in the mitochondria. So these are the changes that are seen in reversible cell injury. In irreversible cell injury, There is rupture of the lysosomes and autolysis by the lysosomal enzymes. Then there is pycnosis and karyolysis and karyorhexis, that is complete degeneration of the nucleus. There is lysis of the endoplasmic reticulum and severe dispersion and loss of ribosomes. There is formation of myelin figures in the cytoplasm, mitochondrial swelling, and formation of large densities in the mitochondria. Let's compare these ultrastructural changes in the reversible and irreversible cell injury. So in reversible cell injury, in the membrane there are just blebs formation and intramembranous particles, whereas in irreversible cell injury there are defects in the cell membrane and formation of myelin figures. In reversible cell injury there is cellular swelling, whereas in irreversible cell injury sometimes the shrinkage of the cell can occur. In mitochondria, both in reversible and irreversible cell injury, mitochondria are swollen. But in reversible cell injury, the amorphous or small densities are seen in the mitochondria, whereas in irreversible cell injury, large densities and vacuoles are seen in the mitochondria. The endoplasmic reticulum is swollen and there is dispersion of the ribosomes in reversible cell injury, whereas in irreversible cell injury there is lysis of the endoplasmic reticulum and severe dispersion of the ribosomes. Coming to lysosomal changes, in reversible cell injury there is autophagy by the lysosomes, and in irreversible cell injury the lysosomes rupture, releasing the lysosomal enzymes which cause further damage to cytosolic organelle. In reversible cell injury the cytoskeletal elements are aggregated, whereas in irreversible cell injury they are disrupted. In reversible cell injury, the nuclear chromatin is clumped or it gathers together, whereas in irreversible cell injury, there is severe damage to the nucleus and it goes through different stages of pycnosis, karyolysis, and karyorhexis. In pycnosis, the nucleus becomes very small and compact because of clumping and severe gathering of the chromatin. In karyolysis, there is disintegration or fragmentation of the nucleus, and in karyorhexis, there is severe fragmentation and loss of the nuclear chromatin. Now let's see light microscopic and gross features of reversible and irreversible cell injury. In light microscopic features of reversible cell injury, there is hydropic swelling of the cell, that is, there is accumulation of water inside the cell resulting in swelling of the cell. Then there may be vacuolar degeneration or foamy bubbly appearance of the cell. There may be accumulation of the fat which is called as fatty change, and this is typically seen in the cells which are dependent on fat metabolism. Example is hepatocytes and myocardiocytes. There is increased eosinophilia of the cell, that is increased pink discoloration of the cytoplasm. Grossly, in reversible cell injury, because there is accumulation of the water, the organ will look paler and there will be increase in the size, the weight and turgor of the organ. In irreversible cell injury, when we see these cells under the light microscope, we will see increased eosinophilia, which is because of protein disintegration and accumulation, which will be more as compared to reversible cell injury, glassy appearance of the cell, vacuolated or moth-eaten type of appearance of the cell, and that is because of digestion of the cell organelle by the lysosomal enzyme, resulting in formation of holes in the cytoplasm. There may be secondary changes which include deposition of calcium in the cells or necrotic tissue, and nuclear changes which include pycnosis, karyolysis, and karyorexis, which can be appreciated on light microscopy as well. The gross appearance of irreversible cell injury takes several patterns, like what we had seen in the previous picture of myocardial infarction, where there was deposition of whitish necrotic area or pale areas. There may be reddish necrotic areas as well. This is the photomicrograph which is showing differences between the normal kidney tubules and the cellular changes that are seen in reversible and irreversible cell injuries in kidney tubules. So this is the normal kidney tubule where there is maintained cell membrane and this nuclear contour and chromatin. In reversible cell injury, we can see that there is clumping of the chromatin and mildly increased eosinophilia of the cytoplasm. In irreversible cell injury, we can see there is disintegration of the cells and there is disintegration and pycnosis and karyolysis of the nuclei, increased eosinophilia of the cytoplasm and membrane disintegration. So this photomicrograph indicates irreversible type of cell injury. So to summarize, a cell can undergo different changes depending upon various stimuli and stresses to which it is exposed. A normal cell, which is in a state of homeostasis, when it is exposed to stress or increased demand, it undergoes adaptation. When it is exposed to injurious stimulus, it undergoes cell injury. If the cell is not able to adapt, then also it will result in cell injury. Depending upon the type of stimulus, whether the injurious stimulus is mild or transient or severe or progressive, will determine the final outcome. If the injurious stimulus is mild or transient, it will result in reversible type of cell injury like cellular swelling and fatty change. If it is severe and progressive type of injury, it will result in irreversible cell injury and ultimately cell death by either necrosis or apoptosis. Necrosis is disorganized cell death and apoptosis is programmed or organized cell death. In the level of adaptation, if the stress is removed, the cell will go back to its normal structure and function. Thank you for visiting my channel PathQuest and watching the video. Please like, share and subscribe to my channel for more quality educational videos on pathology.